Good morning, all of you. Today I'm going to say about the last class I given an introduction about the bioinformatics. So those bioinformatics, what is the tools we are used for the bioinformatics? Actually, we know that all we have we are regularly working with the search engines. The search engines means we have in our hand, we have a mobile phones, with our laptop we have it. So everyone will work with your search engines. Best example I can say that Google search engines, Yahoo search engines. So every providers, means search providers means that's called as website we can call as. Basically URL, universal resource locator. This URL will be gives the search engine. So this search engine works with your location, what your data you saved in your institution or maybe website, you call as website, we can say that will be displayed in your software form. It will be software form. If we can locate your information. So best example, regularly we use it IRTC. So like this we have every RTC bus, RTC, APPSC. Then each and every institution they have their own website they have it. So these websites. But basically suppose when you want to, I'm saying about the biological databases. Last class I said you about that one. So here in this one, I'm going to discuss about what is search engines present in our biology. So what is meant by biology? Biology means living cells. Living cells means we are said to the proteins and nucleic acids. So in our bioinformatics, basically we have one databases we have it. That's called as National Center for NCBI. What is that? National Center for Biotechnology. So these NCBI will have a different subunits we have it. So means hyperlink documents it will provide it. It will provide all the information related to our biological molecules, especially nucleic acid, proteins, genes, then journals and citations, publications. So everything it will be provided. It's a big databases. It's called as big by one. So here, this each search engine. This search engine will work your sequences. It will search our sequence or where you have it. Suppose I am doing a research work in a particular protein. So I got a sequence. Otherwise, I am doing research work on a particular gene. So those genes have sequence, right? I have been isolated from the particular cell. After the isolation of the cell, I have gone for the identification of gene sequence by using the PCR. Everything I did it. So finally I got a sequence. But how could you know that my one is correct or not? So I need to predict that one. I need to confirm it's my sequence is good. I mean my sequence is 100% accurate or not. So for these keys we have a different search engines we have it. So NCBI basically provide that one. So this NCBI will have a different home page we have it. This home page will provide the different logins they have it, like that resources, chemical analysis data, DNA databases, RNA databases, domains, structures and gene maps, homology, and then literature, protein literature, sequence analysis and taxonomy and training tutorials and variations. And it will also provide the login details. And also it will give the particular, we can deposit your sequence, we can retrieve your sequence, we can learn the many things from your NCBI. So I don't want to discuss about the NCBI now. So it's a big databases. So but here, so I'm going to only discuss about the search engines in your biology. It means search engines in bioinformatics. So basically, in our syllabus, syllabus means we have in APPSC and UPSC. They are given us two topics. They are given two topics. The important topics they call as BLAST. So BLAST means basic local search. What is that? Basic local alignment searching tool. So these BLAST will work with our different types of BLAST we have it. So one is a nucleotide BLAST, T BLAST, N BLAST, transcription BLAST. So these BLAST we can use it. Then other one is FASTA. That's called as fast alignment searching tool. So this BLAST and FASTA we can type in your URL. So suppose you want to type your BLASTA, blasta.ncbi.nih.gov./blasta.cgi. This is a URL we can use it. So these URLs they are called as universal resource locators. So each and every institution that's called as URL or maybe domain they have it. Domain or maybe IP address I can say that. Internet protocol, otherwise transition protocol. So this translation protocol will look at your mobile phone, will look at your what is that in your institutional ID, where it will be located, where it will be placed in your institution. So now I am going to discuss about this URL will look at your types of your search engines also. So suppose if you are going to Google Chrome, we have Google Internet. Now Internet we are not in Internet Explorer is not working now. They have removed that one. Explorer till 11 they have used that one. Now each and every Google, Yahoo, what is that, Microsoft, there are people, they are developing the new technologies. New technology means new software developing. So these new software, best example, we can go with the Edge. Edge is developed by Microsoft engine. So here the Blast, I am going to say about the Blaster, basic local search, basic local alignment searching tool. So what will works it is one. So what will simply it will search your sequences. So what is here? So simply when you go to the protein blast, here I am going to discuss about the protein blast. Here we have different types of blast, we have it. So these are protein blast. So what you're going to do, it means here, so when you're going to paste your sequences, so this is nothing but a software. Your blast is nothing but a search engine, is software. So we need to give your sequences. You need to provide your sequences in your search window. So they'll give the specific search windows like that. Entry query sequences, that's called as you need to give your sequence, what your research you did it. Suppose it's a DNA, I'll go with N blast. Suppose it's a protein, I'll go with the T blast. So here they give this space of this one. So these are inquiry query they'll got that. So in this inquiry query we are going to deposit, we are going to paste your sequence what you got from your research. So after pasting that one, otherwise we can upload your file directly from your desktop or maybe web. What is the top you are using? Laptop or maybe desktop you are using. So we can directly paste your sequence. But here when you are going to paste your sequence, we can use the different file formats. We can have it. So basically we know we know that about what is that Word and PDF. What is a PDF? Word means it will be, we can type it in something to those one. So PDF means it's a portable document file. It will be precise. We can't alter that one. We can say that. So the HTML file, what is that, Excel files we have it, table formats we have it, different types of formats we have it. So here, so in this blast we are going to use the plain sequence format, plain sequence test. So suppose myself, I am in Srinivas, so I can use my own sequence. I can design my own sequence format. Suppose we know that language people means that is called as software people, they design that one. So language, suppose Oracle, Java, C++, C++, wherever you have it. So we need to go with the different languages. So your computer will work only based on your languages only. So when you have the command only, your computer will go that. It will work it. Otherwise your computer will be to get crashed down. Crashed down means program will be demounted, means otherwise it will be demolished. So this program will not run, your computer will not run. Like this here also, see your Blaster. This is called as a basic local searching alignment tool. This search engine, we need to give a sequence. It may be, suppose my sequence is protein, I can go with protein Blaster. My sequence is nucleic acid, I will go with nucleic and Blaster. So suppose in my translated sequence, I will go with T Blaster. TB Blaster. It's like this, we have 4 types we have it. In this one, so only I want to give the brief idea about the Blaster only, clear? So here, this sequence has to submit that one here. So in this Blaster, we have a phases we have it. Means, so how you going to deposit that? How you going to submit your sequence? So first one is, what is that, entry query sequence. That will have a space we have it. In this space, we need to give that one. So they'll also ask what is the job title. What is the job title? Now what next they'll ask the align, they'll ask how many sequences you want to align it. Align means putting in a sequence.